This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 260, recorded on February 24, 2022. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schechter.
Well, hello there. Hello, everybody.
How's everything? It's 72 and sunny there?
72 and sunny. It's not 72, but 68 and sunny. But complaining it's too cold.
That's so funny. Also joining us from Charleston, South Carolina, Michael Schmidt.
Hey, everyone. Spring is sprung here in Charleston. We have the famous Chamber of Commerce weather. It's 81 degrees or 27.2 Celsius, and it's a glorious day. The sun is out, there's a light breeze, no humidity, and the birds are singing.
Well, here in New York, it's freezing. It's still winter,
Yes.
which is fine by me. I have a nice coat.
Yes.
I want to tell you about the Department of Microbiology and Immunology at Louisiana State University Health in Shreveport. They would like to encourage people interested in pursuing graduate studies in the fall of 2022 to apply to their master's and PhD programs. They have a vibrant department of 16 faculty members working on a variety of DNA and RNA viruses, innate immunology, and intracellular bacteria such as Bordetella and Legionella. Located in northwestern Virginia, they offer unique opportunities to grow personally and professionally, milder winters, and southern hospitality. Deadline for applications, March 1. We'll put a link to the department for your perusal. Ever been there, Michael?
Yes, I actually have. One of my classmates used to be on faculty there before he defected and went to work for NIH, I should say. He left the academic route, and he's now an SRA for the NIH. It's a great place.
I was scheduled to visit in February 2020.
Uh-huh. We know what happened then.
Yeah. Actually canceled. I mean, they wanted me to go, but I said, no, I don't think so, because things were just blowing up.
Yeah, and you were in ground zero being in New York City, so you would have literally brought the virus with you.
Indeed. All right, today we're going to talk about food, which is a good thing that I've just eaten my lunch and I'm not going to get hungry. We're going to start with apples and Elio.
Okay. An apple a day keeps you-know-who away. And then that's the idea. So here's the story. Apples are among the most consumed foodstuff there is. Okay. And very little is known about their microbiology. Because when we eat apples, we eat bacteria and fungi. And so analyzing the apple microbiome to detect difference between tissues and the management of apples, organic and conventional, and what happens after you harvest them, all of that makes a difference and can be studied by using for bacteria the 16S-RNA gene amplicon and qPCR for fungi. So they are also using fluorescent in situ hybridization and confocal laser scanning microscopy. So each apple fruit harbors different tissues. An apple has a stem, peel, fruit pulp, seeds, and underneath a thing called the calyx. And they're colonized by different bacterial communities. The hotspots are the fruit pulp and the seeds. The peel is less colonized. Surprising, it's the outside. You would think you would get a lot. Anyhow, for a gram of apple, they found about 100 million 16S RNA gene copies. So this was not influenced too much by management, but some. despite the similar structure in general, the bacteria that dominate are proteobacteria, about 80%, Bacteroidetes, 9%, actinobacteria, and Firmicutes make up the rest. And so some of these are health affecting and so could be enhanced by by management, the apple afterwards. So we consume about 100 million bacterial cells per apple. Okay, that's a lot of bacteria. It sounds like in weight, it's not. So the study looks at the main factors defining the fungal and bacterial diversity and the composition of apple fruit. Post-harvest treatment and temperature storage have a strong impact on both fungal and bacterial diversity. The change with different post-harvest management practices. The greatest impact, they say, was attributed to sanitation practices, with major difference among unwashed, washed, and washed waxed apples. I guess we all know what that is. Sometimes you get an apple and it looks like it's coated with wax. Well, it's because it's coated with wax. The magnitude was tissue specific, with a great impact occurring on the peel tissue. So we learned a lot about this. There's a lot of data here. It's not surprising exactly. I wouldn't say that this is something that startles you. So that's the story. That's the story. I can't improve on that.
Elio, the wax makes fewer bacteria or more bacteria?
It makes more.
Why is that?
I don't know. I don't think they don't say. I don't know.
The other interesting tidbit was when they compared European apples to North American and South American apples, and then the ones from the Middle East, they found distinct microbial differences, which really puzzled me, because you would think an apple is an apple is an apple. Yeah. But it goes back to some of our earlier discussions about terroir, the French word I can never pronounce, about the importance of the local environment and influencing wine varietal production and how you can have good wines from certain regions. So I'm wondering, as I was reading this, and this is an absolutely fascinating paper because I didn't know anything about the Royal Gala apple. I didn't know anything of how it came about, and the fact that most of the apples are out of this New Zealand cultivar, and it's actually an interesting read. I was reading the paper originally at lunch as I was munching on an apple, so it may have been just a little bit more interesting because I was eating the apple and I was wondering about this fruit I was eating. And it was a Walmart apple, so I didn't know whether or not that made any difference. But
I like the Galas. Those are my favorite.
Yeah.
Apparently, a lot of people like them because it's a big portion of global production.
Yes,
it's very interesting. Now, Elio, they talk about pathogens of apple. Are any of the bacteria that identified known pathogens?
Not really. I don't think so. I don't remember they saying that. By the way, I didn't tell you that the article appeared in Environmental Microbiology. It's called Global Analysis of Apple Fruit Microbiome. Are all apples the same? The answer is no. Okay. And the authors are from Austria, about 20 authors. Austria, Sweden, Israel,
your alma mater, Vincent, Cornell,
West Virginia, China,
Turkey,
Cornell,
Uruguay,
Ontario, Canada, Nova Scotia, Canada, Italy, Spain, Turkey, Switzerland, Uruguay, and Austria. I think I said Austria. So they're from all over the place. This is a big effort big international effort as well. It should be, because different apples in different countries have different microbiomes.
Do these, so we eat an apple, we get a lot of bacteria. Does that influence our microbiome in our intestines at all?
They didn't do that. They didn't study what the effect really is on people. But I would think it'd be fairly minor. I mean, although we eat a lot of bacteria when we eat an apple, 100 million bacteria is not a lot of bacteria. So I wouldn't think.
So, Elio, if you could grow apples without any bacteria, what do you think would happen?
Nothing.
It would be fine. The apple would taste the same and all that.
The apple would taste the same. I don't think it would make any difference.
But if you were to use that apple to make cider, it would probably taste different.
Sure. Well, the yeast on the apple will count when you do that.
Yeah.
It's totally true. But I don't think that the effect of bacteria on people is going to be very marked. Do you think? What do you think, Vincent?
I don't think it's going to have much of an effect on us unless, for example, you've just had an antibiotic therapy and you're restoring your microbiome. Maybe if you ate apples, it would help to restore it. I don't know. But I suspect the bacteria, as Michael said, probably contribute to the taste, right, of the apple. It might taste different.
Maybe. I mean, most of them are on the skin. You know, there are very few in the pulp.
Yeah, because once you get into the pulp, you'll begin to consume all the carbohydrate and sugars, right, and you'll begin to rot the fruit, and that's what results in spoilage. So I can understand why they did this study, because it's so important to store the apples, because apples are harvested principally once a year.
Yeah.
And yet we have apples year round. It's one of those fruits that we have. And, you know, it's interesting that they went with both the Northern Hemisphere and the Southern Hemisphere, because Uruguay is effectively, you know, it's the equivalent of New York State apples from South America. Mm-hmm. And it was actually interesting just comparing and contrasting, looking at these things. And, you know, the New Zealand digression of that they were involved in the production of the original Gala. It made for a fascinating read, and it's an interesting time as we begin to explore more.
Michael, didn't we do a paper from Joe Handelsman years ago on the apple orchard microbiome?
Yes. I was trying to remember when it was, and I have to go back and search, and we'll maybe put it into the show notes.
That's must be related to some extent, right?
Yes.
So they're stored for up to a year. Wow.
Yeah.
That's quite something.
Well, that's why they wax them and the cold storage. Yeah. Whenever I buy apples and put them in the refrigerator, they seem to rot just as fast as when I leave them on the counter.
Well, maybe they're a year old. That's the problem.
That's the problem.
Anyway, do you think it would be an interesting experiment or useless to grow apples without bacteria?
The notobiotic apple.
Yes, the notobiotic apple.
The notobiotic apple.
My dime is on the fact it would make no difference.
Make no difference? Yeah, you're probably right. I do like that, the notobiotic.
The notobiotic apple.
Yeah.
Why not? It wouldn't be hard to do.
Let me see. TWiM. Let's search for TWiM apple orchard microbiome.
Handelsman.
Joe Handelsman did that, and let's see. It's got to be Handelsman. Her student did it. Where is Joe these days? In Wisconsin, right?
Wisconsin. She's at Wisconsin.
It doesn't come up right away. I have to.
I'll go look for it, and then I'll stick it in the show notes.
Ashley Shade. Remember? Ashley Shade was the student on it. Unexpected diversity during community succession in the apple flower microbiome. It's,
my neurons are resurrecting it.
That's MBio, and that one was published. Well, why don't they put the year right on the front page, Michael?
I'm not in charge.
2013. So this must be the one I'm thinking about.
Yeah,
but it doesn't look like
we talked about it.
But we should.
We'll put it in the show notes.
Put it in anyway, because I remember that meeting at ASM, and she was there. Ashley was there, and I said, "Oh, we should talk about this on TWiM, why don't you come on and do it sometime? And she said, yeah, sure, I'd be happy to. And then I never followed up. All right, there it is in the show notes.
It'd be nice if somebody did a similar study with pears, because in my mind, apples and pears are kind of similar. They're not, but they feel like they belong to the same group of fruits, don't they? Anyhow,
yeah.
Except so pears rot faster than apples. They don't have this long of a shelf life, and pears are picked more green. And if you ever notice the difference between a ripe pear that you pick from a tree yourself versus the one you buy at the grocery store that was picked before,
I see.
This whole issue of fruits, we should probably think about getting a real food person to help us navigate some of these things sometime.
Well, food is a big subject, right?
Oh, absolutely.
Fruit is just a part of it. There are tons of fruits. Apples,
pears. And it's full of microbiological tidbits.
For sure.
Yes.
You got pears, you got apples, you got peaches, nectarines. What else?
Tomatoes.
Well, what grows on a tree?
Okay. What grows on a tree? Plums, apricots.
Plums, apricots. Cherries? Yeah, cherries.
Trees.
I mean, there's a lot of...
And I would guess that those microbiomes might be somewhat similar being in a tree environment as opposed to something closer to the ground like a strawberry or a tomato, right?
Right.
And strawberries are quite fragile. I mean, you just look at them sideways and the fungi are taking them over.
Strawberries, raspberries, blueberries.
Blackberries.
Oh, man. And then,
It's a good thing you ate lunch, Vincent.
It is. I was hungry. I got to the incubator. I have some food stored in the fridge here. I got yogurt, so I had that. But, I would be
Did it
hungry.
have any fruit in it?
Yeah, dried cranberries.
Oh, that's good.
I love dried cranberries, yeah. I put them in my yogurt. And I also had some pecans in the yogurt, too, today.
Well, that's good.
Anyway, speaking of food, Michael, you have something to tell us about food?
Oh, yes. Keeping with our theme, why did the bee eat the chicken? That's the beginning of the title of this paper. Why did the bee eat the chicken? Symbiote gain, loss, and retention in the Vulture bee microbiome. And this paper is by Figueroa, Macaro, Krikchilsky, Yangnaga, and McFrederick. And they are from 5 different institutions: the Department of Entomology at Cornell, the Department of Environmental Conservation at University of Massachusetts Amherst, the Department of Entomology at UC Riverside, the Department of Ecology and Evolution at Columbia University, and finally, the Division of Invertebrate Zoology from the American Museum of Natural History. I believe that listeners of TWiM would likely spot me that a significant change to diet can alter the microbiome of the host, and that the microbes that we harbor allow us hosts to pull nutrition from novel foods. However, what I did not know is that bees are simply wasps that switch to a vegetarian lifestyle. This was news to me.
I didn't know that.
This was news to me. And that the vast majority of bees are vegetarian. They feed on pollen and nectar. So in this paper, we are going to learn some remarkable facts about bees, principally the stingless species. So this is a bee that doesn't have a stinger, that collect carrion. So instead of not having a stinger, they actually literally slice flesh off a carrion and have fully reverted to a necrophagious lifestyle. That means they eat dead things to get their carbon and energy. So they rely on carrion for protein, forgoing what I always imagined bees do, their traditional visitation ritual to flowers to collect pollen and nectar. What is even more remarkable is that this study was done in the lush jungles of Costa Rica, where what I would have assumed they had plenty of nectar and pollen. So these bees are termed...
Tusk bees.
Yes, these bees are termed vulture bees, belonging to the corbiculate aphid clade, which is known for its ancient association with a small group of core microbiome phylotypes. So today's story is going to take us down a path of investigating what makes the vulture bee microbiome tick, along with closely related facultative necrophagous bees. So these are bees that eat carrion and then can also rely on nectar and pollen. And then the obligate Carnivorous species, which only eats pollen and nectar. And they take us on this journey to understand the diets and how the diets then influence the microbiome structure of the bee. So I'll admit, I spotted this paper in January when many of us are still trying to adhere to our New Year's resolutions. To see what perspective that this might offer, as many of us went going meatless Monday, alcohol-free Januaries, with some of us toying with veganism, vegetarianism, or our normal routine of just being an omnivore. So I'll admit that I try to keep this TWiM exercise interesting for our regular listeners and to really try to entice new ones. So I thought this paper might be able to help us think about how changes to our diets might be studied and interpreted. And we have 3 distinct classes of bees that we're looking at that are eating unique substrates. We have a bee that only eats carrion, the obligate necrophage. Then we have the ones that can go both ways, the facultatives. And then, of course, we have the completely vegetarian version, which I would have always imagined as a bee. So what did they do? Well, as you might imagine, this is going to be a story very similar to the last one that Elio told that involves deep sequencing of the 16S ribosomal RNA gene as our way of getting into learning about the community of microbes that are affiliated with the vulture bee. And then via statistical methods, we are going to try to decipher the consequences of this behavioral switch and its relationship to the vulture bee's core microbiome. The bottom line is that they learned that the vulture bees have lost some of their core microbes, retained others, and then entered into a novel association with acidophilic. These are microbes that make acid as their principal waste product found in the environment, as well as the acidophilic microbes are coming from the carrion they were eating. So the authors then present us with a hypothesis that the abundance of the acidophilic bacteria from their survey suggests to them that the acidic gut is important for the vulture bee's nutrition and health, as has been found in other carrion feeding animals like the human race. So it's also not surprising to me that the facultative necrophagous bee has a more variable microbiome. So I would toss us into that category. Those of us who experience with meatless Mondays, we need a much more diverse microbiome than if you only eat meat or you only eat vegetables. And that's effectively what they found, suggesting that the bee diet may interact with the microbiome of the bee on both a short and long-term time scale. So if you take this back and ask, how does it matter to us? Understanding these crazy bees may actually help us think about meatless Mondays and ask yourself, do we need to be worrying about the consequences of our behavioral changes to our diet? And then we now know that the microbiome is so important for everything in terms of general health. Will it impact our health? And none of those studies, I think, have ever been done. So this is our first foray into beginning to think about how to do that sort of science. And, that's me dropping the gauntlet to my next generation of scientists. You got to think cleverly of how do you know whether or not meatless Mondays is a good thing for you? Well, that's off the topic of the paper, so I'm going to get us back on track. Otherwise, Michelle will be cross with me. So the authors take us on a remarkable journey, educating us about this category in B. And in fact, One of their references is from the time of Linnaeus, with a citation from 1758. And Linnaeus was the one who alerted us to one bumblebee species that feeds on carrion, Bombus terrestrius, from 1758. And then there's another that say...
Well, Linnaeus came later.
Well, it's from 17... They said 1758 in the citation. So I took them at their word. And then Say in 1837 talked about Bombus effetatus, which also fed on carrion. But the obligate necrophagy has been found in only 3 closely related Trigonia species in these neotropic regions like Costa Rica. They found it in Trigonia hypogea, necrophagia, and crass bees.
These are what again?
These are vulture bees.
Okay.
These are bees in the aphid clade. So enter their first figure, where they show us their sampling strategy. They had 32 bait stations, and the feeding behavior, they actually show us what looks to me like a breast of chicken, literally covered with bees, as the bees are literally slicing a piece of that chicken meat off for themselves, as opposed to coating themselves in pollen. They're literally coating themselves in chicken meat. And they take us on this journey through Costa Rica. First, what I want to offer is due respect to these authors. They present a lot of tedious work. They collected 159 pollinivorous, facultatively necrophagic, and obligately necrophagic Melipone bees from 9 genera and 17 species from these 32 bait stations from La Selva and Las Cruces field stations in Costa Rica. And they give us a map of where they did this in Costa Rica. And it took me on my winter vacation going to Costa Rica as I was looking at this paper. And when I read it originally, it was January and it was cold here in Charleston. On the carrion baits, they found 9 Melipone species from 4 genera, and 3 species on chicken baits were novel for them being facultatively necrophoric. Now, I really liked the color coding of their figures. It was consistent throughout the paper, though I do have one quibble. They actually depicted necrophagy as being blue, and I would have actually depicted necrophagy as being red, simply because I consider when you eat meat, there's always blood. So I would have thought they would have coded that red, but instead they coded it blue. The vegetarians were coded red, and the facultatives were in between as their greenish orange color. And I can never tell the colors because I'm color defective. So they had to make sense out of the tremendous amount of data that they collected. And I said this was a heroic effort. They collected approximately 13 million 16S ribosomal RNA gene amplicon reads as they collected from these 32 base stations. So the depth of coverage for the average sample that they collected was approximately 74,000 reads. And from a perspective of deep sequencing, that is very deep. Talk about depth of coverage. This is really pretty deep. And even after they did the quality control, chimera removal, decontamination, removal of the chloroplasts and mitochondrial reads, they still offered us pretty deep sequencing in that each sample that they report on had over 14,000 reads. Why does this matter from the 172 samples? Is you know that the microbes that they're reporting are really likely all there. They're just not missing any of the microbes. So what did they learn from all this work? Well, not surprising was that the species of bee was highly significant. It was highly significant for the predictor of alpha diversity. And so now we need a diversion to reacquaint those listening what alpha diversity means. It's defined simply as species richness associated with the sample. So you have a lot of species in that particular sample. So the species of bee was a predictor of species richness, and that the diet was still significant, but only marginally so after they controlled for the bee species, which, the way some folks deal with that is with Shannon diversity, where they will combine richness and diversity. And recall that Shannon diversity factors in the number of species and then talks about the inequality between species abundance in the sample that you are reporting. But they didn't do that, so that was a minor quibble. They also offered that, and this is a converse view, that the collection substrate was not a significant predictor of their amplicon sequence variants that they observed. And they attribute this likely to the fact that the facultative species were found across a collection of substrates. The facultatives didn't care where they were getting their food from. The facultative necrophagic bees are sort of like graduate students. If there's food out, they'll eat it. Well, it's true. If there's food out, graduate students can become omnivores or vegetarians. They'll eat anything. And so this further highlights the importance of the species of bee and the diet. And I really liked their figure. If you look at the second figure in the color coding, the vegan bee, or the one that only eats pollen and nectar, really didn't have all that much diversity. It was hovering around the mid-30s. But then when you look at the facultatives, it's not surprising that they had much more diversity in the microbial species, again, attributing to the diet. The obligates, on the other hand, were somewhere in between the vegan and the, if you will, omnivore. The next figure in the results was very interesting to me when you look at it. It almost appears like a Venn diagram. And they offer through their presentation, and this is something I had to go look up because I never was familiar with this. It's non-metric multidimensional scaling ordination, or NMDS. And NMDS is an indirect gradient analysis approach, which produces an ordination, think X and Y axis, based on the dissimilarity. And to me, that's why it looked like a Venn diagram. You had the vegan bees, you had the carrion eating bees, and then you had the facultatives. And when you look at figure 3, you see the color coding is again the same. And the bees that are present, and when you look at the microbes that are present, and you get a sense of the microbial community composition by the host and species diet. It was, to me, a really easy way to look at that, and at the same time, it made sense. Because if you look at the obligate, you can see they're clustered in one zone. And if you look at the absent ones, they're clustered in another zone. And then you have some that are tossed in the middle. And so here they offer that the composition of the bee gut microbiome and the beta dispersion also differed by diet. And it was a really nice way to look at it. And then when you begin to think about beta diversity, it's the difference between different samples, because alpha diversity is within a sample, and then beta diversity is looking between the others. They report on the microbes that are present, and it really is fascinating. But what the next figure enables us to do, and this is one of the greatest figures that I ever seen in order to compare and contrast differences between types of microbes. So on one scale, you have the obligate necrophage, the facultative necrophage, and the vegan bee. And that's on, if you will, the y-axis. And then across the x-axis, they have the bacteria that are involved. And then in the heat map designation, where the darker the color, the greater the proportion that that species is represented. And so like Snodgrassella, which is in the vegan bee, and as its name implies, Snodgrassella would be associated with pollen.
Yep.
And indeed it is. And it's the most abundant species. So this was a really easy figure. And this is the average, the title of the figure is the average relative abundance of the corbiculate core, the corbiculate associates, and environmental bacteria by host species and diet. And so the corbiculate core, which is the core microbiome of a bee, and then those microbes that are associated with the bee's microbiome only transiently, and then the environmental acidophiles that are effectively coming from the carrion or outside of the environment. It was a really great way of presenting a lot of complicated data where you could actually figure out what was going on. So it was really quite remarkable. The species from a single specimen was captured, and they actually reported on that species. And what they finally conclude is the acidophilic microbes that had been found in the environment or on meat were more significantly more abundant in vulture beetles, while the corbiculate core bacteria were more significantly abundant in the obligate pollinivores. And that was principally Bifidobacterium and Snodgrassella. So what they learned from all of this work is reversion to a carnivore lifestyle in the obligate necrophage involved retention of some of the ancestral core microbes, as well then as acquisition of these new lactic acid bacteria and acetic acid bacteria. The extreme diet switch from just eating pollen and nectar to actually going to carrion was likely facilitated by or resulted in the novel composition of the vulture bee microbiome. No big surprise there. The third fact that should not be surprising was the new microbiome is likely to provide a novel function to the host, or as they offered in the discussion, we know that our stomach, the acid in our stomach, provides the mammalian carnivore a diet enriched in amino acid digestion pathways, while the mammalian herbivore microbiome are enriched in amino acid synthetic pathways. And again, this is no great surprise to anyone who ever took a Van Niel or Stanier-like course in microbial discovery, like some of us who were classically trained did, where we went out to various soil types and asked who's there. They conclude their discussion of the importance of the acquisition of new lactic acid bacteria and acetic acid bacteria and helping the bees adapt. And previous listeners to TWiM will appreciate that these two categories of microbe, the lactic acids and the acetic acid bacteria, are important symbiotes for insects, especially bees. For example, lactobacilli that colonize bumblebee guts can inhibit the growth of a gut fungal pathogen via the lactic acid production. And in fact, we discussed that very fact on episode 245 of TWiM, and I dropped that reference into our show notes. And while they did not measure the pH of the gut pH of the bee, the dominance of the acidophilic bacteria in the vulture bee gut suggests that the acidic gut environment is important for these bees as well. So summing things up, they took us on an excursion to Costa Rica, where they presented sequence evidence that reversion to this carnivoric lifestyle in a bee to an obligate necrophagic form of life had profound consequences for its microbiome. They offered that their observation that the enrichment of the acidophilic bacteria in the vulture bee requires further additional studies and also functional studies to ask, what is the acid doing for the bee beyond the obvious of preventing a fungal bloom. They end by offering to the reader that these fascinating bees offer us rich insights into how diet interacts with gut microbiomes so that as we lose our battle with our New Year's resolutions, we might better understand what we are doing to our microbiomes as we go meatless Mondays or alcohol-free Januaries.
Michael, is this these bees are only in Costa Rica? Is that why they had to go there?
Yes.
Okay.
They're unique to Costa Rica.
Interesting. So what's the ancestral bee? Is it a pollen eater or a carnivore? Do we know?
I assume that these bees effectively are an evolution of wasps that went completely vegan.
I see. So, wait, wasps, what did you say about bees in the beginning? Bees are wasps that switch to a vegetarian lifestyle. Okay. So wasps are carnivores.
Right.
So all wasps are carnivores? Is that right?
No, only some. Only some. And I'm not an entomologist, so I'm sure we'll get letters from entomologists, but forgive me on that. But I found their work to be rigorous and fascinating, and the deep sequencing was sufficient to convince me of their conclusions.
So basically wasps became vegetarian bees, and then a few of them decided they liked meat, so they went back.
Yes, they went back.
Okay. This is not for me.
This is not for me. I'm going to go back or the competition for the nectar, because remember, Costa Rica has a vibrant bird population and they have lots of hummingbirds. So my guess is it's competition.
Mm-hmm.
It's probably
they found another niche that they could survive in. Yeah. So has anyone compared the microbiome of either vegetarian or meat-eating humans? That would be interesting, right?
Yeah. Well, I mean, especially as we can change our diet willy-nilly.
Sure.
And everyone is talking about probiotics and the need for probiotics. And I don't think we know the consequences of some of the decisions we are making.
Well, there are people who study that. I remember there's a guy who talked at ASM once.
Yeah.
He said in Japan, you eat a lot of seaweed, so you get bacteria that can digest the seaweed for you, right?
Yeah, and we actually did that paper on TWiM too, I think. So it's
so, Elia, what do you think of this one compared to the apple one?
Well, this is a little bit easier to swallow.
Very good.
And I think it's a little bit broader. The apple thing has a certain predictability to it in terms of study.
Well, I think they're both interesting. Yeah,
it's a little bit more challenging.
Yeah. All right. Thank you, Elio. Thank you, Michael. That'll do it for TWiM 260. Show notes are at microbe.tv/TWiM. If you can send us a question, comment, TWiM@microbe.tv. And if you like what we do, consider supporting us financially, microbe.tv/contribute. Elio Schechter is at Small Things Considered. Thank you, Elio.
My pleasure.
And Michael Schmitz at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.